Welcome back. In the next two web lectures, we're going to be taking you on a very brief tour of the different parts of the cell and the different functions that cells carry out. And in so doing, we'll put together a picture of why the cell is the basic unit of life and how an individual cell carries out all the functions that we've been talking about that are necessary to consider something alive. According to cell theory, the cell is the fundamental unit of life. So all organisms are made of cells, and the cell is the simplest collection of matter that can be alive, the smallest thing, the simplest thing that can perform all of those functions that all living things do. All cells are related by their descent from earlier cells. What does that mean? That means that there is a single common ancestor that gave rise to all cells that are alive today, including all the cells that make up your own body. And we believe this because although cells can differ substantially, they're going to share common features that are present in every single cell alive on Earth, from unicellular bacteria all the way up to blue whales. So first of all, let's think just very briefly about how we know what we know about cells. What are the methods that biologists use to study cellular form and function? So if we first think about form, what are cells made of? What's inside of cells? Obviously, microscopy is a very useful tool to be able to magnify these cells and be able to see the cell shape, the cell structure, and even inside the cells to look at the organelles. So in a light microscope, visible light is passed through a specimen and then through a glass lens. So those lenses are then going to bend the light and magnify the image so that it's visible to the human eye. We can also have a different kind of microscope, electron microscopes, that instead of passing light through the specimen is going to shoot electrons at it. These electron microscopes are used to study subcellular structures. And the two basic kinds are scanning electron microscopes, so they're going to focus a beam of electrons onto the surface of a specimen, and that's going to create images that look three-dimensional. And to examine the interior structures in a cell, we can use transmission electron microscopes, which are going to focus a beam of electrons through a specimen to be able to see the internal structures. In terms of the functions of cells, being able to understand what the cells are doing and what the different parts of cells are doing, biologists use a method called cell fractionation, which is going to take cells apart into their component organelles, so usually using some kind of blender to homogenize the cells, and then by using a centrifuge at different speeds and for different lengths of time, you can separate different components of cells with different weights and densities and sizes so that you can isolate individual organelles to be able to experiment on them and see what they're doing. The book goes into quite a lot of detail on this. If it's interesting to you, feel free to read that in depth. But basically where I want you to be with this is understanding the tools that we use and how we gathered all this information that we're going to be talking about what's in a cell and what those different things do. So it's important to know how we know what we know. So just as an overview of the different sizes of things that we can observe with different methods, so if we're talking about whole organisms, there are some very large nerve and muscle cells, such as the squid giant axon, that can actually be seen unaided. Chicken eggs, obviously, we can see perfectly fine. Frog eggs are getting to be close to the smallest things that we can really study without the use of these microscopy tools. So in the range of light microscopy, we have things like cells, human egg cells. We can see the nucleus just fine with a light microscope. Most bacteria, there's some very small ones that can't be seen with a light microscope. And even going down to the larger organelles, such as mitochondria, we can extend this range using enhancement techniques, which are described in the book, to see things even smaller. But generally, once we get down to the subcellular level, we're talking electron microscopy to see such small things as viruses, large proteins, and then below what we're even able to see with an electron microscope are things like water molecules. Nobody's ever actually seen one. Small molecules and atoms. So now let's start taking a look at how these cells are put together. So the concept 6.2 in your book is eukaryotic cells have internal membranes that compartmentalize their functions. Before we focus so exclusively on eukaryotic cells, we'll talk a little bit about the differences between prokaryotic and eukaryotic cells. So the basic structural and functional unit of every organism is one of two types of cells, either prokaryotic or eukaryotic. So only organisms in the domains bacteria and archaea consist of prokaryotic cells. So remember the three great domains of life, bacteria, archaea, and eukarya. Protists, fungi, animals, and plants are all eukaryotes. So these are all part of that third domain, eukarya, and they consist of eukaryotic cells. Based on morphology or form, cells can be divided into these two fundamental types based on the presence or absence of a membrane-bound nucleus. So eukaryotes have them, prokaryotes don't have them. But it's important to keep in mind that based on phylogeny or evolutionary history, the organisms are actually divided into those three domains, bacteria and archaea, which are both prokaryotic, and eukarya, which is eukaryotic. And the reason it's important to bear this in mind is that archaea and eukarya are actually more closely related to each other than either is to bacteria. So being a prokaryote doesn't necessarily make you part of the same phylogenetic lineage. You'll learn much more about that kind of evolutionary tree thinking when you get to the second semester of BioCore. So let's compare prokaryotic and eukaryotic cells. First of all, we said that there are basic features of all cells. This is why we can have high confidence that all of these cells are related by common descent. They all contain proteins that perform most of the cell's functions. That's uniform throughout all of life. They use nucleic acids to store, transmit, and process information. And these nucleic acids are packaged into chromosomes, which carry the genes. They use carbohydrates as a chemical energy source and a source for carbon, structural support, and identity. They use a plasma membrane that's composed of lipids that serves as a selectively permeable membrane barrier. Remember, that's important for this idea of homeostasis, having an internal environment that's separated from and can be different from the external environment. They have a semi-fluid substance inside the cell called cytosol. More on that as we go. And they also all use structures called ribosomes to manufacture their proteins. So let's think a little bit about these prokaryotic cells first. So they contain a single circular chromosome. Again, a chromosome is a single DNA molecule, and that chromosome is composed of that one DNA molecule and associated proteins. The chromosome contains genes, which code for polypeptides, which we folded into active proteins. And the chromosome is very tightly coiled to fit inside the cell. The actual circular chromosome, if you were to stretch it all out, it would be much, much, much larger than the cell. And it's sort of localized in a region of the cell called the nucleoid. It's not membrane bound, it's not physically separated, but it tends to be found in a particular location in the cell. They also may contain small, separate DNA molecules called plasmids. So again, these are circular and much smaller. And we usually think of these prokaryotic cells as being very, very simple, much less advanced than eukaryotic cells. But recent discoveries have shown that prokaryotes are actually more complex than we thought. So let's see some examples of prokaryote complexity. So prokaryotes also have ribosomes. These are sophisticated macromolecular machines that consist of RNA molecules and protein and are used for protein synthesis. They contain these structures shared by all cells, but they also, some of them, have internal photosynthetic membranes. So these are not chloroplasts. These are membranes within the cell folds of membranes that increase the surface area for collecting sunlight to conduct photosynthesis. They develop from folds of the plasma membrane, so they're going to be continuous with this outer plasma membrane, and they're going to have green chlorophyll to conduct photosynthesis. And so this is really important in the history of life. If we look at the entire history of the Earth on this sort of circular clock-like scale, so up here at the top would be the origin of the Earth, we get the formation of the moon slightly thereafter, and going all the way around, when we come back to the top again, that'll be the present time. The first life begins around here. If we think of this as a 24-hour clock, we'll start to put times on here, but remember this is a 24-hour clock rather than the 12-hour clock you're used to looking at. So we start to see photosynthesis here right around 6 o'clock in the morning, if this is our geological clock here. And this is in bacteria, basically. This is where we start to get photosynthetic bacteria. It's not until about 1 o'clock that we start to see eukaryotes at all. So this is when we get the first eukaryotic cells, 1 o'clock in the afternoon. Getting late in the day already, by 3 o'clock, this is when we first start to see the very first multicellular organisms, so well into the afternoon. And it's not way until up here at 10 o'clock at night that we get the first green land plants. So bacteria were way, way, way eons ahead of plants in terms of photosynthesis. They've been doing it almost forever. So in addition to these photosynthetic membranes, some bacteria do have membrane-bound compartments, and we can call them organelles since they are membrane-bound. And these organelles are going to perform specialized tasks, including storage of ions that are needed for their physiological processes, containing magnetite crystals to help with orientation and finding their way through an environment, organizing enzymes for building organic compounds. One of the great benefits of having these separate membrane-bound chambers is that you can hold all of the molecules that you need for a particular biochemical pathway close together and keep that biochemical pathway running smoothly. So now let's think about eukaryotic cells. So eukaryotic cells are characterized by having DNA that is contained in a nucleus that's bounded by a double membrane. Eukaryotic cells also typically have membrane-bound organelles. In eukaryotes, we can say that having membrane-bound organelles is the rule rather than the exception. In prokaryotes, it's the exception rather than the rule. It happens occasionally. And then they have a cytoplasm, which is defined as the region between the plasma membrane on the outside and the nucleus. So everything that's within the cell but outside the nucleus is considered the cytoplasm. Eukaryotic cells are generally much larger than prokaryotic cells. So here you have an example of an animal cell and a plant cell, both of those eukaryotic, and then the average size of a prokaryote here next to it for scale. So generally much larger cells. And we know that the metabolic requirements of a cell set an upper limit on the size a cell can be. This is an incredibly important topic. I'm not giving it much time here, but I'm actually going to post a separate web lecture explaining in detail, step by step, this idea of surface area to volume ratio and why it's so important. So stay tuned for that. But long story short, as a cell increases in size, its volume grows disproportionately more than its surface area. So surface area provides the nutrients that are needed to the volume, but the volume is getting much larger, much faster than the surface area. Please check out the other web lecture for the details on that. So the first cell structure that's common to all kinds of cells is the plasma membrane. So this is going to separate life from non-life. We've talked a little bit about this before. We're going to go into very great detail on this just after the unit on introducing the cell. So in short, the plasma membrane is a selective barrier that allows sufficient passage of all the materials needed for the cell. So this is the surface area that's going to be providing materials to the volume of the cell, if we think about those surface to volume relationships. And it's going to be the surface over which waste is going to be removed from the cell. As we've mentioned before when we talked about lipids, the plasma membrane is composed of a phospholipid bilayer and is embedded with these proteins that can either span the entire thickness of the bilayer or attach to one side or the other. Again, it's very important in homeostasis, creating a distinct internal environment that's going to be regulated under a narrow range of conditions that are optimal for the life processes that the cell is undergoing. Distinct internal environment, a stable internal environment, regardless of what's happening in the external environment. The plasma membrane makes that possible. As we just mentioned, inside the membrane, all of the contents of the cell are collectively called the cytoplasm, excluding the nucleus. So everything between that plasma membrane and the nucleus we call the cytoplasm. If we think specifically about eukaryotic cells, a eukaryotic cell has internal membranes that divide the cell into compartments or organelles. So this is the endomembrane system. So organelles break up the large cell volume into smaller membrane-bound organelles. This compartmentalization offers two big advantages. First of all, you can contain chemical reactions and keep them away from other possible chemical reactions that are going on that could interfere with each other. Also, you increase the efficiency by making sure that all of the chemicals that are needed for a particular reaction or pathway are there in close proximity to each other. So that's going to make this reaction a lot more efficient. The molecules aren't just floating freely anywhere in the cell. You've got them all close together where they need to be. Plant and animal cells have mostly the same organelles. There are some that are particular to one or the other, but there's some uniformity between plant and animal cells. And so here we can see a diagram. This is the same one you saw a couple of slides ago, showing an animal cell with a nucleus, with chromosomes in it. We see structures such as a mitochondria and a Golgi apparatus, endoplasmic reticulum, a cytoskeleton, and we see the same things in the plant cells. We see a nucleus containing chromosomes, endoplasmic reticulum, Golgi apparatus, mitochondria, plasma membrane. But as we'll see, particularly in the next web lecture, plants also have these very special organelles called chloroplasts. They also have this large central vacuole that's lacking in animal cells. Animals have their particular organelles called lysosomes that are missing from plant cells. So the next main concept is that the eukaryotic cell's genetic instructions are contained in this membrane-bound nucleus. And these genetic instructions, the instructions for creating polypeptides, which will then be processed into functional proteins, is executed by the ribosomes. They're the actual structures that construct these polypeptides. So the nucleus contains most of the DNA in a eukaryotic cell, but not all of it. In the nucleus, the DNA is organized into discrete units called chromosomes. So each chromosome is a single DNA molecule, and its associated proteins called chromatin. So most of the time in the cell cycle, the chromatin just kind of looks like a diffuse cloud if you visualize it, except when that cell is about to divide, and that's where it condenses down into these discrete chromosomes, which are these little structures with the center and the tails on each side that we're used to thinking of as chromosomes. So the ribosomes use the information from the DNA to make proteins using an RNA intermediate. The RNA is the unit that actually travels out of the nucleus to where the ribosomes are. So the nucleus is usually the largest structure that you see inside the cell, and it's highly organized. So it's surrounded by a double membrane called the nuclear envelope, which is studded with little pores. So you'll see these little openings. Those are the pores where the messenger RNA is transported out of that cell nucleus to send those instructions to the ribosomes. And the inside surface is linked to a structure composed of the cytoskeleton. You'll learn more about that in the next web lecture. And this is called the nuclear lamina, and that is a sheet of these fibrous proteins, part of the cytoskeleton that maintains the shape of the nucleus. The nucleus has a distinct region called the nucleolus, where ribosomal RNA is synthesized and the ribosomal subunits are assembled. We'll talk more about what ribosomal subunits are as we get to ribosomes. So here we have some images of what all of this looks like. Here is transmission electron micrograph showing the little bumps. These are the little pores in the nuclear envelope where those messenger RNAs are going to come through. And we see in this image here the double membrane of the nucleus. The outer membrane is continuous with the endoplasmic reticulum. More on that structure in just a moment. And here are all these little pores, which are actually composed of these protein compounds forming a donut shape called a pore complex. And this is what they look like in electron micrograph. Here's a close-up of that nuclear lamina, this meshwork cytoskeletal structure that's going to maintain the structure of the nucleus. And here we have the chromatin, which has the DNA molecule kind of coiled around these proteins that keep the chromatin somewhat structured within the nucleus. What are these ribosomes? So basically they're protein factories. These are, I like to think of them as sort of the workbenches where these polypeptides are put together. This is where those condensation or dehydration reactions happen that attach the amino acids to each other in just the particular sequence. So we'll learn more about the details of how that happens when we get to translation a little bit later on in the semester. But for now, ribosomes are complexes made of ribosomal RNA and protein. They lack a membrane, so technically they're not considered organelles. Some of the ribosomes are just freely floating in the cytoplasm or the cytosol, and these free ribosomes are going to manufacture proteins that are intended to be just distributed throughout the cytoplasm or that need to go to other organelles. So things that are going to stay close by or just kind of be diffused throughout the cytoplasm. Some ribosomes are attached to the endoplasmic reticulum. So these are going to manufacture proteins with other fates that are not going to be just dumped into the cytoplasm. These are going to be packaged and sent somewhere. So looking at an image, the ribosome comes, as we said, in two subunits. There's a large subunit and small subunit. More about the function of these subunits when we get to translation. Here we have an image of what these different kinds of ribosomes look like. The freely floating ribosomes just kind of look like little dots floating in the cytoplasm. But when you see a membrane with those little dots attached, those are the ribosomes that are attached to the endoplasmic reticulum, a molecular structure of large and small subunit of the ribosome. So now let's take a look at how the endomembrane system works as a unit to regulate protein traffic and performs metabolic functions in the cell. So the endomembrane system consists of the nuclear envelope, so that double membrane surrounding the nucleus, the endoplasmic reticulum, which we just started to look at, we'll look at in more detail in a moment, the Golgi apparatus, lysosomes, and vacuoles. And these components are all either continuous with or connected via transfer by vesicles. So vesicles can bud off of one membrane and then join with another membrane later. And so all of these membrane systems are connected to each other, either physically or by this budding off and joining up. So now let's take a look at the endoplasmic reticulum. The endoplasmic reticulum is an organelle that's an extension of the nuclear envelope. So you can think of this outer nuclear membrane. Remember, there's a double membrane. The outer one, just think of it as expanding out, proliferating wildly, and creating all of these folds of membrane. So all these folds are going to be continuous with that outer membrane of the nucleus. So there are two types, rough and smooth. The rough endoplasmic reticulum is the part of the ER that has those ribosomes bound to it. So these ribosomes are going to manufacture the polypeptides, and these are going to have a carbohydrate attached to them, covalently bonded to the protein. This rough endoplasmic reticulum is also going to distribute these proteins in transport vesicles. So the proteins that are intended to be secreted to the outside of the cell are going to be surrounded by these membranes. They're going to fuse with the plasma membrane of the cell and open up and release those contents into the area outside the cell. It's also a membrane factory for the cell, so this is constantly producing new cell membrane. The ribosomes in the rough ER are going to synthesize proteins that will either be shipped to another organelle in a very specific way, so we want it contained a little membrane vesicle to keep it all together until it gets to that other organelle. These could be proteins that are going to be inserted into the plasma membrane, so be part of those intrinsic proteins in the plasma membrane. So all it has to do is stick those proteins in the membrane that it's budding off. When that membrane joins with the plasma membrane, those proteins are already there in that cell membrane and they just become part of the cell membrane. Or, as we just discussed, secreted into the cell exteriors. So as proteins are manufactured on the rough endoplasmic reticulum, they're going to move into the lumen. So the lumen is the inside of this sac. If you were to follow where this is connected to the nuclear envelope and go sort of to the inside of this sac, there's kind of an interior and an exterior. And the interior part is the lumen. So it's the inside of any sac-like structure. Inside the lumen, the proteins are folded and processed. So this is where they take their functional form. And these proteins that are made to be packaged in these little vesicles, they may be proteins that are going to carry messages to other cells. So those might be the ones that are kind of dumped into the space outside the cell to go and carry their messages to other cells. They might act as membrane transports or pumps, so those intrinsic membrane proteins, or they may be something that's going to catalyze reactions, and then we want to keep them contained within a vesicle, so they only catalyze the reactions where and when the cell needs them to. So the smooth endoplasmic reticulum lacks ribosomes, and in its lumen it contains enzymes that catalyze reactions involving lipids. So these reactions may synthesize lipids needed by the organism, so this is going to be part of that function of working as a membrane factory to actually put together these large complex macromolecules. It can also have enzymes that are going to break down lipids and other molecules that are poisonous. And breaking down lipids is a very important first step to cellular respiration, being able to use the chemical energy contained in those lipid bonds. It's going to start the process of metabolizing carbohydrates. It's going to be a reservoir for calcium ions, which you'll see over and over again as you go through general chemistry. Calcium ions play many, many different roles in physiology and lots of different organisms. So this is a very important function. So now let's look at the next part of this endomembrane system, the Golgi apparatus. So we can think of the Golgi apparatus as sort of the shipping and receiving center. So it's going to work very closely with the rough endoplasmic reticulum. So the Golgi apparatus also consists of these flattened membranous sacs, and we call these membrane-bound sacs cisternae. So what does the Golgi apparatus do? It's going to modify the products of the endoplasmic reticulum. So it's going to take those folded polypeptides and further modify them. It's also going to manufacture certain macromolecules, so it's going to have some synthesis functions. It also sorts and packages the materials that come from the rough ER into transport vesicles that are going to take them to the specific location where they need to go. The Golgi apparatus also has a distinct polarity or sidedness. So there's a cis side that is facing the nucleus, and then there's a trans side that's facing the plasma membrane. And so different things are going to be happening at the cis versus the trans surface of this Golgi apparatus. And so what are the functions? Processing, sorting, shipping proteins synthesized in the rough ER. The cis side of the Golgi apparatus receives the products from the rough ER, something's going to happen to those products while they're in the Golgi apparatus. They're going to be modified and sorted. On the trans side, they're going to be repackaged into vesicles in the sorted form. So they're going to be sorted into different kinds of proteins with specific destinations, and those will be packaged into very specific vesicles that are going to travel to very specific locations. So here's an image of the Golgi apparatus, and you can see it just looks like these flat membranous sacs. So again, we're looking at a slice through. This is more of a three-dimensional view, and you can see that it's lots and lots of membrane and these internal chambers, this internal lumen, with a cis side facing the nucleus. So this would be the cis side here, a trans side facing the plasma membrane. So let's put all of this together, what we've done so far, and see how this would actually work. And so the best model that we have right now for how materials move through this endomembrane system is what we call the signal hypothesis. So according to the signal hypothesis, all proteins start to be synthesized on free ribosomes. So they're going to start to synthesize their protein. The proteins that are bound for the endomembrane system are going to have a zip code somewhere in their amino acid sequence. And so we're just going to call that a zip code. It's going to basically identify that protein as something that has to be shipped somewhere. So that's going to direct the growing polypeptide to the rough endoplasmic reticulum. That ribosome will attach, and that's those ribosomes attaching to the endoplasmic reticulum is what makes it the rough endoplasmic reticulum. It's not fundamentally different in any other way from the smooth endoplasmic reticulum. So the zip code consists of a 20 amino acid sequence, and we call this the ER signal sequence. So that is the sequence that indicates that that particular protein needs to be taken to the endoplasmic reticulum. So then the ER signal sequence binds to a signal recognition particle, or SRP, and those two together move to the rough ER membrane and bind to a receptor there. So this is what's going to actually connect the ribosome with the growing polypeptide to the endoplasmic reticulum. At that point, once it's bound to the ER, that signal recognition particle is then released, and then the protein synthesis continues. But instead of the protein growing just kind of loose in the cytoplasm, that protein is going to be growing into a channel or a pore that goes into the lumen of the rough ER. So that growing protein is fed into the RER lumen, at which point the ER signal sequence is removed. It's not part of the functional protein. It's just a tag that indicates that it's for export. So looking at this in diagrammatic form, we've got a free ribosome in the cytoplasm. We've got the mRNA attached. It's being read by the ribosome and starting to create this polypeptide chain, the green part. And then as part of the polypeptide chain, it produces this ER signal sequence. So this is part of this growing polypeptide that's coming out of the ribosome. So that is going to attract this signal recognition particle. It's going to bind to it, and that's what's actually going to connect it to the endoplasmic reticulum, to a receptor on the membrane of the endoplasmic reticulum, which is also connected to what's called a translocon, which is the pore or channel through which this growing polypeptide is going to be fed through. Once this is all bound and this process is started, the ribosome is now sitting on the translocon and feeding that polypeptide chain down into the lumen of the ER. The SRP is released, the SRP receptor dissociates from that translocon, and everything is up and running. Once it's inside, the polypeptide chain is modified so that ER signal sequence is removed. Again, it's not part of the protein, it's just a name tag or a zip code, and then the protein is now in the lumen of the rough ER, where it's going to be folded and processed and ready to ship out to wherever it's going. In the lumen of the rough ER, the proteins are folded. The enzymes within the lumen add carbohydrate side chains in a process called glycosylation. So the carbohydrates are going to serve as another indicator that this is to be shipped. And that resulting glycoprotein is ready to be shipped to the next destination. And that next destination is going to be the Golgi apparatus. So they're transported in vesicles. The vesicle, remember, is just a little bubble of membrane that buds off from the ER. It's going to move away and fuse with the membrane of the cis face of the Golgi apparatus, that side facing the nucleus. It's going to fuse with that membrane and dump its cargo to the inside, the lumen of the Golgi apparatus. Inside the Golgi apparatus, the protein sequences all have their little carbohydrate tags. Okay, so proteins bound for different destinations are going to contain different carbohydrate molecules that are going to tag them for their specific destinations. So these are floating in the lumen of the Golgi apparatus. And these proteins are sorted. How is this done? So different areas of the trans side of the Golgi apparatus is going to have a concentration or a cluster of receptors for particular attached carbohydrates going to a particular destination. So the ones with the orange tags, for example, are going to be attracted to these orange tag receptors that are all concentrated together in a specific part of that membrane. And so that's how they're sorted. The yellow ones are going to bond with these receptors that are all concentrated in that area. And similarly, the purple ones are going to be attracted to this area with their own receptors. And then when those parts of the membranes bud off, they're going to bud off with just proteins that are meant to go to that particular destination. So for example, the yellow tags might tell it to go to another organelle. The orange tags perhaps are telling it to go to form a lysosome. We'll learn about the lysosomes in just a moment, but the hydrolytic enzymes that are needed to form a lysosome, and then maybe the purple ones are going to the plasma membrane for secretion outside of the cell. All right, so another important part of this whole endomembrane system, and we'll see how this all works together. We'll put it all together into a big single picture at the end, but are these little membrane-bound compartments called lysosomes. So these are basically digestive compartments. So we can think of these as the recycling centers. They're going to make sure that when different organelles get old, their materials are broken down and reused to create other macromolecules that are going to make new organelles. So they're going to contain approximately 40 different enzymes. So that's where these vesicles coming from the Golgi apparatus to lysosomes are going to carry those enzymes into these developing lysosomes. And they're going to digest macromolecules and export the monomers to the cytosol to be reused to build other macromolecules. So the enzymes are acid hydrolases. So these are going to work best in an acidic environment, again, emphasizing the importance of these membranes that separate these enzymatic activities from the rest of the cell. The rest of the cell doesn't want to be at pH 5. And the way it gets that pH is that it's got proton pumps intrinsic in the membrane that pump protons into the lysosome and maintain that low internal pH. Another little membrane-bound structure that your book mentions in conjunction with the mitochondria, because it is associated with metabolism, but I'm going to put it here just because it is connected to this whole endomembrane system and works as part of the same functional unit. So peroxisomes originate as buds from the ER, and they are a center for oxidation reduction reactions. So one of the purposes of these reactions is to break down things like fats or other large macromolecules into smaller units that can then be used in the mitochondria to break down further to provide energy for the cell. But also they have enzymes that can oxidize different toxins in the cell, such as the peroxisomes in liver cells that break down ethanol in alcoholic beverages. They're going to keep those kind of separated from the rest of the cell. There are also specialized plant peroxisomes called glyoxysomes that oxidize fats to form an energy storage compound. And these Oxidation reduction reactions often produce hydrogen peroxide, but then that hydrogen peroxide is also toxic. So in peroxisomes, there's also an enzyme catalase that detoxifies it and processes that hydrogen peroxide back into water molecules. The final component of this endomembrane system I want to familiarize you with are vacuoles. So these are a diverse category of just separated little compartments surrounded by membranes for various purposes. So vacuoles are large membrane-bound vesicles derived from the endoplasmic reticulum and Golgi apparatus, found primarily in plants and fungi. So what are their functions? Some are specialized for digestion, so they're going to be digestion reactions going in there, breaking down large macromolecules. Most are used to store water and ions to help the cell maintain its normal volume. So that's the main function of this enormous central vacuole that we see in plant cells that take up most of the space in the cell and sort of squish all the rest of the cytoplasm kind of between the central vacuole and the cell membrane. In seeds, they're generally filled with proteins that are going to be needed as the plant begins to grow. In flower petals or fruits, they're going to contain the pigments. And they also can contain toxins to help protect the plant from herbivores. Finally, if you've ever looked at something like a paramecium under a microscope, you might see this sort of pulsating structure in there. And what that is a contractile vacuole. It's basically pumping excess water out of the cells, and this is needed in freshwater organisms because they're maintaining a saltier environment within their cell than their external environment. So water's going to tend to come in, it needs to be pumped back out. So now let's put this all together in a final review, integrating all of these different components of the endomembrane system. So the endomembrane system is a complex and dynamic player in the cell's compartmental organization. These membranes are morphologically the same in all of these different compartments of the endomembrane system and also in the external plasma membrane, and they're constantly budding off from one membranous structure, joining with another one. So this membrane is being constantly recycled between these different structures. So collectively, lysosomes, Golgi apparatus, and ER are going to be important components of the endomembrane system to produce, process, and transport proteins and lipids. And as an example, if we think of the acid hydrolases that are used in lysosomes, we can say that their instructions are stored in the nucleus. They're going to be transferred out of the nucleus, out to the ribosomes, where they're going to attach to the ER and be synthesized directly into the endoplasmic reticulum. They're then going to be sent to the Golgi apparatus, where they're going to be processed and sorted and shipped. In this case, we've got a vesicle that's going to fuse with what's going to become a lysosome to provide those enzymes that are needed for the reactions that occur in lysosomes. If we think about the way these membranes kind of work together in the system of just a lysosome, so some types of cell can engulf another cell by phagocytosis and form a food vacuole. So here's an example. This cell membrane just pinches in. Again, we'll talk about this more when we get to cell membrane structure and function. It's going to surround this food particle in a little piece of the membrane that's going to bud to the interior of the cell as a food vacuole. This lysosome is then going to come and its membrane is going to fuse with the membrane of the food vacuole and join its contents together. So these digestive enzymes are going to combine with the food and break it down so that those monomers from the macromolecule can be used by the cell to build other macromolecules. But a lysosome can also use its enzymes to recycle damaged or old cell organelles. So these cell organelles that are marked for destruction for whatever reason, for recycling, become enclosed in a vesicle. That vesicle, the membrane, is going to fuse with the membrane of the lysosome, again releasing its digestive enzymes to break down those defunct organelles into their component monomers to use again to build into other macromolecules. So this is sort of the big picture of this very continuous membrane system with all these little bits of membrane that are being constantly reused, separating, rejoining something else. So a big question that's raised by all of this is, how do these membrane-bound vesicles know where to go? How do they get to their destination? This and many other very fascinating questions are going to be answered in the next web lecture, where we look at mitochondria, chloroplasts, the cytoskeletal system, and also the world just outside of the cell.